All right, 12s. Continuing on with our discussion of the kidney. Today we're talking about urine formation. How is urine formed in the kidney? Last time we talked about the structure of the kidney, and we talked about there's our kidney here. Blood coming in via the renal artery, and then into renal capillaries. Then it gets smaller and smaller, afferent, arterial, and we've made it to the small microscopic working structure of the kidneys, which is the nephron. That's this. This is the nephron. And the nephron is surrounded by blood vessels. So, how is urine formed? So remember, we're filtering blood here, and the filtering essentially happens immediately. It's called pressure filtration. So you get pressure filtration here. So at the afferent arteriole, you enter the peritubular. This is called the glomerulus. And this section here is called the Bowman's capsule. And so what happens here, the blood comes in and it's under high pressure, and these, the capillaries in the glomerulus are leaky. And so small molecules under this pressure just shoot out of the glomerulus and are absorbed or go in to the Bowman's capsule. And that's it. The filtering's done. We're done. Except we're not done, because the problem is everything basically that's small gets dumped. So what you're getting dumped, what's getting filtered out here is water, some water. You're dumping out ions. So those are like important, calcium ions, sodium ions. They're important. They're basically salts. What you're dumping out is salts. You're dumping out your food monomers, so glucose, nucleic acids, all those things that you did all that hard work digesting in your digestive system, which we talked about, that were absorbed in the small intestine. Now you are dumping it out into the nephron. And along with those things, you're also dumping out urea and uric acid, which is those things that you don't want. Now, these things are now outside of your body, technically. So because if you look, this network, these tubes, they continue on into the collecting duct, and then out into the renal pelvis, out the ureter, and then out the ureter to the bladder, and out through the urethra, out into the air. So basically, at this point, the inside here of the Bowman's capsule is continuous with the outside world. And that's why you get bladder infections, because the outside world can get in through that hole, urethra, and so bacteria can get in and cause bladder infections. People get bladder infections because they got a dirty urethra, and it's inside your body, but it's actually outside your body. It's crossed a membrane now. All those things outside your body. So, amino acids, glucose, nucleic acids, lipids, all those things, all those small things are now out here. Those things that you want, and urea and uric acid are also out here. Things you don't want. So now what's going to happen is you are going to bring those in. So just to note, the blood here now is different than the blood here. So the blood in the afferent arteriole has glucose, has all those amino acids, urea, uric acid, all those food monomers. But the blood here in the efferent arteriole has less water, has less ions, so those salt ions and stuff. Food monomers are gone. So when I say food monomers, I mean all those things, those products of digestion. and urea and uric acid are gone. And so now what's left here are red blood cells, platelets, white blood cells, and globulins like albumin and all those things. So that is that's what's left. And basically, because we've lost some water, this still has a greater concentration. It still is basically hypertonic to the afferent arterial. It's like thicker blood. Okay? Here's another image of that Bowman's capsule, and you can see, so it's basically, it's like it's like a little hollow tube with the blood vessel going in, and the blood vessel leaks out in, and the stuff gets pushed down into the proximal tubule, water and stuff. So, okay, so now we have to reclaim all those things that we've lost. They're out in the outside world. We work so hard to bring them into our body, the food, the glucose, and all those things. We worked so hard in our digestive system, as we learned, and now we've dumped them out. So now we have to bring them back. And where we bring a lot the products of food digestion back is we reabsorb them. So it's called selective reabsorption. And where it happens is in the proximal convoluted tubule. And I will show you where that is. So basically, the proximal convoluted tubule is this section here. It's right after the glomerulus. Let's see if we've got there we go, proximal convoluted tubule. So there, it's before this thing called the loop of Henle. This is the proximal convoluted tubule. So there's blood vessels all around the proximal convoluted tubule, and there's integral proteins all over here, channel proteins that are selectively pulling in. So they're pulling in the glucose, they're pulling in some of the ions, they're pulling in the amino acids, the nucleic acids. What they're not pulling in is that urea and that uric acid. We don't want that. Those things can continue on and continue out. Next, so here's a beautiful image of it's in the notes. So glucose, amino acids, so sodium, and so water are being reabsorbed back into the blood in the proximal convoluted tubule. What's left in the blood, you can see here, is cells and albumins. That's what's left in the blood. Wonderful. Now, what happens next is rather complicated. Do we have a nice, there it is. There's the lovely image of the loop of Henle. So we've reabsorbed our amino acids and stuff, and now our job is to pull in water. So we've dumped, remember, a lot of water here. We've dumped a lot of water in the we've dumped a lot of water in here from the glomerulus into the Bowman's capsule. So this is all full of water, and we're reabsorbing. And as we enter the loop of Henle, this heads from the cortex into the medulla. Remember, there's the cortex, and this is the medulla. And I said the medulla is going to be salty. It's going to be salty because kind of a crazy thing happens here. What happens here is salt, so back here, salt from the filtrate is being actively pulled out, okay, by proteins here, the transport proteins, so it's using ATP, which is making the area around the loop of Henle salty. It's making it hypertonic. So there is a concentration gradient. And what happens is where does water go? Water heads to the area that's hypertonic through osmosis. So through passive transport, this part of the loop of Henle is covered in what are called aquaporins. They're basically, they're like channels for water. And so because we've transported all of this salt out into here, what happens is through osmosis, the water leaves the loop of Henle and it leaves the filtrate. So by the end here, the filtrate is super duper concentrated. You can see here, this is its osmolarity. So it's the highest concentration at the bend in the loop, basically. So some water is going to come back now because it's so concentrated, except there are no aquaporins. Okay. So what happens is, so you got salt here. Water is being pulled out, so there's a great concentrate. Now you're pulling out the salt and you're making it more diluted, but so there's just a little bit of water, and that water has now been diluted of salt. And this thing kind of fuels itself because the salt is being pulled out here. The water osmoses out here, which makes this more concentrated, which makes salt come out there. So water comes out here, salt comes out. This is called the descending limb, and it's where water passively osmoses out through channel proteins. And this is the ascending limb, where salt is pumped out actively through channel proteins. This pumping makes the area around the filtrate hypertonic, which powers the water osmosing out there. And so now what you've done is using this loop of Henle, which is actually really long. This is not to scale. It goes all the way down. It goes way down, if you imagine. It goes way down into the medulla. And what you've done is you've reclaimed your water now, or a lot of your water, and you've reclaimed the ions that we lost. A lot of those ions that we lost. So here we're reclaiming the products of digestion. Here we're reclaiming water and salt. Okay? So that's all the stuff that's happening just to make your urine. And remember, there's millions and millions and millions of these nephrons working all inside. This is a microscopic structure inside your kidney, and millions of these are working to filter your blood. Okay, next we have to talk about the distal convoluted tubule. So here's the distal convoluted tubule. And what happens here is a couple things are going on here. So there's a blood. We've reabsorbed water and we've reabsorbed salts. And the kidneys, the kidneys, its job is to get rid of urea and to get rid of uric acid. The other job is to regulate the pH of the blood. A lot of pH regulating happens here. And so what happens in the distal, number one, this does not have to do with regulating the pH, is drugs are excreted here, so are transported across. And this includes things like histamines, vitamins like vitamin C and C. So these would be large molecules that cannot exit via filtration out of in the Bowman's capsule. So they have to be actively transported out. So like a big thing is, if you eat a lot of vitamin C, for example, people do blasts of vitamin C. I'm gonna do a blast of vitamin C to improve my immune system. Well, immediately what will happen is your body senses that it's got too much vitamin C, and so it will blast that excess vitamin C right out here. It'll transport it out, and there it is. It's outside of your body. So you paid a lot of money to eat a lot of vitamin C, and it's basically just traveled from your stomach, from your digestive system, into the blood, and then immediately out into your urine. So, I don't know, eat an orange instead. And okay, so back to regulating pH. Is there a better image of regulating pH, or is that my best one there? Is that my best one? That looks like that's my best one. Okay, so what happens at the distal convoluted tubule is you have bicarbonate and hydrogen ions. And basically, depending on, so if your blood, if the pH is too low, so it's acidic, if your blood is acidic, what will happen is hydrogen ions will be pumped out, will go out, and bicarbonate will be reabsorbed in to raise your pH and make your blood more basic. Remember, your blood's pH has to be around 7.35. That's what your body's aiming for. If your blood is too basic, what will happen is hydrogen ions will not be pumped out, and bicarbonate will not be reabsorbed back. Is that right? Hopefully I'm right. Yeah, that's it. So that's how it adjusts. Either it adds more hydrogen ions and absorbs HCO3 if your blood is too basic, and alternatively, if the pH of the blood is too high, so your blood is too basic, less hydrogen ions will be excreted and less HCO3, so bicarbonate will be reabsorbed. So we're reabsorbing bicarbonate to raise the pH to make the blood more basic and excreting H+, or we're not excreting H+ and not reabsorbing bicarbonate. And so then by this point, the blood has the correct pH here as it's leaving now the peritubular network, and it's going out. It's in the venule now. It's going back to be pumped into your systemic system. It's been filtered, and so it's got the right pH. It's reabsorbed. It's got all its food things back in it. The concentration of vitamins is correct. The water volume is correct. There's still some water volume that's going to be done here in the collecting duct, and basically what happens here in the collecting duct is ADH, a hormone called ADH. What does ADH stand for? Diuretic hormone. ADH. Anti-diuretic hormone. There we go. Anti-diuretic hormone. And basically what ADH does is it opens water channels here, so water is reabsorbed out of the collecting duct. ADH effect. There we go. So water will be reabsorbed back into the blood, so that even more water gets reabsorbed. So we got this whole loop of Henle system to reabsorb water, and we've got the collecting ducts. And ADH can be interfered with, and we'll talk about that next time. So there you go. So know how urine is formed and what things are reabsorbed. What happens in that Bowman's capsule, right? So you've got the afferent arterial and the efferent arterial. So afferent arterial blood. Know the difference between afferent arterial blood and efferent arterial blood, right? So what's the difference between this blood and this blood, right? Everything here has been dumped out. And then as it travels through the proximal convoluted tubule, glucose, amino acids, some salts, water are reabsorbed. So this is selective reabsorption here. Then you've got this whole osmosis system here, right, where we're pumping out salt into the area around, into the medulla. So it's really salty. And what's going to happen is water in this part, water is going to, because the area around is hypertonic, water is going to try to equalize the concentration. So there's an osmotic gradient, and water moves to the area that's hypertonic. And so we've reabsorbed salt here, and we've taken away a lot of water. Then here we're secreting drugs and anything that we want out of our body. Large molecules that we want, hormones, drug, histamines, things like that are being excreted here. And also pH, the blood of the pH is being settled here by excreting hydrogen ions and absorbing bicarbonate to make the blood more basic, or vice versa, not excreting a lot of hydrogen ions and not reabsorbing bicarbonate to make the blood more acidic, depending on what the needs are. And then finally, in the convoluted tubule, we're dumping out more water. We're pumping out more water. This area is going to be, a little bit salty. Now, some urea. Oh, we need to talk about. So the urea concentration is super high here. At this point, right? Because we dumped out all the urea, we've dumped out all the salt, and we're dumping out all the water. So actually, some of the urea leaks out here, and it makes this area hypertonic. It adds to the hypertonic state of here, and then which promotes water coming out of the collecting duct, and the urea will actually diffuse back along the collecting duct. The urea will diffuse back here in the ascending limb. So it's kind of urea moves through here. It's moving backwards. I don't know if that makes sense. Right, so let me just rephrase that. Let me just say that urea stuff again. So because the concentration of urea is so high, it will actually diffuse out some of the urea here in the collecting duct. It will contribute to this area being hypertonic, which contributes to water coming out here, osmosing out of the descending limb, because there's lots and lots of aquaporins here, so lots and lots. So this area has lots of channels for water. This area does not, but this area does. And then the urea will be reabsorbed back in the collecting duct. So a little bit makes a sort of circle here, and it contributes to water reabsorption. Okay, I think that gives you an idea of how urine is formed. Anything else I'm forgetting? Anything else? I don't think so. Okay. There you go. That's how urine is formed in the nephron. And then what we'll talk about next time is how that process is regulated. There's not much there in there.